Uh, Good morning, everybody. So first of all, I would like to thank the organizing community to give me this opportunity to present my recent result. So my study mainly focused on in the interactions between the plant, the mycorrhiza fungi, and the fungal hyphae-associated bacteria. So we know for a mycorrhiza plant, there are two P-optic pathways. So one is we call root direct pathway. Another one is mycorrhiza P-optic pathway. So today my talk will mainly focus on this tiny area, maybe around the hyphae. So maybe only 1 millimeter wide. So this tiny area we call hyposphere. So how the phytate turnover in this tiny area. So some years ago, we published a paper. So in this study, we want to understand how to understand the contribution of different phosphate to plant phosphorus nutrition. We used such kind of compartment systems in order to separate the growth space of root and the mycorrhiza hyphae. We used the nanomesh. So this 30 micrometer nanomesh can only allow the hyphae penetrate, but do not allow the root penetrate. So then we can get a root compartment and a hyphae compartment. And then we added the phosphate in the hyphae compartment to look at how much contributions that's give to the plant through the mycorrhiza pathway. So what's the interesting thing we found is the phytate give the highest contribution to the plant after 10 weeks. So we checked that the phosphatase activity around the hyphae. So we concluded that the mechanism is mainly because the mycorrhiza hyphae can produce the phosphatase and mineralize the organic phosphorus. But in 2012, Tisserand and his colleague published their transcriptome research result. In their study, in their result, they find so the vascular mycorrhiza fungi actually cannot produce phytase. So with this new result, we think we should reconsider our previous and reconsider how to explain our previous studies. So in such kind of razor box systems, also we sterilized the soil, but we cannot avoid the environmental contamination, the bacteria contamination from the environment. So we suppose that some bacteria must be involved in the mineralization of phytate. Therefore, we raised two questions. So the first one is there collaboration between AM fungi and the hyphae-associated phosphorus-soluble bacteria we call PSB in terms of mining the organic phosphorus from the soil? The second question is, would EM fungal hyphae exudate drive phytate P turnover and plant utilization? So to answer these questions, we conducted three experiments. In the first experiment, we used such a cultural system. So with petri dishes, so we avoid any contamination, any environmental contaminations of the bacteria. So we inoculated the root with mycorrhiza fungi, and allow or do not allow the hyphae access into the hyphae compartment. And we added the phytate in the hyphae compartment, and we also inoculated the phosphorus-soluble bacteria in hyphae compartment. And what we found is that the bacteria can really colonize on the hyphae. And after 2 and 4 weeks, we found significant increase of bacterial number. That means the bacteria, the fungal hyphae, released the exudate, really stimulated the growth of the bacteria around the hyphae. So that is in this in vitro culture systems. So in the next experiment, we just want to understand in the real soil systems, if the fungal hyphae still can release substantial amount of exudate and feed the bacteria, and the bacteria would drive or promote the mineralization of phytate. So to answer this question, so we use a method we call that's called C13 DNA zip method. So we labeled the hyphoexudate to label the plant by C13 carbon dioxide, and then we checked the carbon flow from the root to the hyphae. And we use this compartment systems. We inoculated the plant with and without mycorrhiza fungi, and with or without PSB. So the PSB, some of the PSB strains that we know they are sequences, and we also added the phytate in this hyphae compartment. So we extracted the DNA from the hyphae compartment, and we separated the carbon 13 DNA and the carbon 12 DNA by centrifugation. And so we really find some of the TRFP fragment was really labeled by the carbon 13. And then we sequenced this fragment, and we found, finally we found one of the fragment. So the sequence is almost 100% similar as the Pseudomonas acaricinas that we inoculated before the experiment, so inoculated into the hyphae compartment. So from this result, we confirmed that the hyphae compartment and the bacteria in the in the hyposphere can really use the hyphoexudate. The hyphoexudate originally comes from plant photosynthesis. So if the bacteria has really worked on the mineralization of the phytate, so that's the result. So we've really found, so in the compare with so the non-inoculated treatment. So if we inoculate bacteria and the mycorrhiza fungi, we really found that the organic phosphorus content was reduced. But unfortunately, in this experiment, we didn't find the plant get any benefit from the mineralization of phytate. So this is plant growth, and this is plant P uptake. So where has the phosphorus gone? So if we check so phosphorus, soil microbial biomass phosphorus, really we found in this double inoculus treatment, so the microbial phosphorus was significantly increased. So this result indicates that there is a competition for available phosphorus between the bacteria and the mycorrhiza fungi. So in this case, in such a P-limited soil, So the bacteria competed with the phosphorus. So that caused the fungi so did not get any benefit. So if we could manipulate the interactions between the mycorrhiza fungi and the bacteria in the soil, so we think that in such a P-limited soil, so the carbon is limited, and the phosphorus also limited. So the bacteria want to use the phytate P. So they use the hyphae exudate, then mineralize the phytate, but they did not release the mineralized phosphorus to the hyphae. So if we can reduce the carbon and the phosphorus ratio by adding some small amount of available phosphorus, what will be happening? That's so our hypothesis is so we could manipulate this process. So if we reduce the carbon and phosphorus ratio, maybe the competition will be not so stronger. So to test this hypothesis, we still use this compartment system. So we inoculate the plant with and without mycorrhiza fungi, and we added a phytate in this hyphae compartment, and inoculate the bacteria also in this hyphae compartment. And so here is and also we added 0.05 milligram per kilogram of available phosphorus to regulate the carbon and the phosphorus ratio in the hyphae compartment. So here is the result. So if we did not added any available phosphorus, so we will find that the result is similar as before. So the phytate was mineralized, and the mineralized phosphorus was given to the microbial biomass, but plant did not get any benefit. But if we reduce the carbon and phosphorus ratio, so we still find the mineralization, but we did not find the further increase of microbial biomass phosphorus. And the plant then, in this case, get the benefit. So the double inoculate treatment, so that is double inoculate treatment have highest phosphorus content compared with the control. So from these studies, so we confirmed that in the hyposphere, so interaction between the mycorrhiza fungi and the bacteria could be competition. But the competition can be shifted to the cooperation, so that's controlled by carbon and phosphorus ratios. So that's my main conclusion. So finally, I would like to thank a lot of people, my students and my colleagues and the funding agents. And finally, I would like to thank you for your attention. Good morning, everybody.